I'm Marcia Fournier, Program Manager of the Gabriella Miller Kids First Pediatric Research Program. Welcome to our fall webinar. I want to start actually talking about our topic of collaboration and what means collaboration for us at the Kids First program. Collaboration, we collaborate to generate data, share data, and analyze the data. And it's a collaboration between the NIH investigators, the Kids First Sequencing Centers, and the Kids First Data Resource Center, and all researchers out there. We collaborate to bring this data closer to doctors, patients, and families. So I invite you to join this community and collaborate with the Kids First program. And actually, I would be curious if you have ideas of how to collaborate with the Kids First program, please add to the chat box your ideas throughout the webinar. I also have a few items that I want to tell you. One is if you have questions, feel free to use the Q&A box, and we will be answering questions throughout the webinar as well. And we will give you a survey in the end of the webinar, and we welcome your answers. We use it to guide us to organize future webinars. I want to thank you, the Kids First DRC, the Data Resource Center, for partnering with the Kids First program and organizing this webinar. I will start with the agenda. I will tell you a little bit what I learned about the Kids First program since I joined as a program manager earlier this year. And then following, we will have opportunity to hear from Kids First investigators about the collaborative research across multiple pediatric conditions. And then we will also listen from the Kids First Data Resource Center about the popular genetic variant browser, which is a tool offered through the cloud. And also the Kids First Data Resource Center will share what we have in terms of support, training, and other resources. So let me start a little bit with something about myself. My education is in cancer research. I came to this country about 25 years ago from Brazil. Exactly, I came from warm Rio de Janeiro in February with 40 Celsius in Rio de Janeiro to Boston below 30 Fahrenheit. Was a cultural shock, but also thank you to that scholarship that brought me to this country to develop my doctoral studies. It kind of changed my professional and also my personal life as I've raised three kids in this country. Throughout my career, I had opportunity to work both in academia and industry. And if you ask me what connect the dots of my career is like a big scientific question of how to use genomic data to answer questions that will help diagnosis, treatments, and prevention tools for doctors, patients, and families. So the Kids First could not be a better fit for me in my background. Data resources that I have worked in the past, usually more often than not, they are targeting one type of data, let's say genomic data, transcriptome, or one type of disease like breast cancer, lung cancer. But the Kids First program, what I learned is that by building a resource that allows multiple data types and also data from across multiple pediatric conditions, it allows a deeper understanding of the biology behind the conditions and also a richer data for researchers to work with. Also, I learned about the commitment of the program to share genomic data associated with phenotypic and clinical data, which again give additional meaning to the genomic data. I was amazed to learn about the history of the Kids First program. All started with Gabriella Miller, who died at 10 years old of brain cancer, and she was a fearless advocate for childhood cancer. And the Kids First program is part of her legacy that she left behind. In 2014, a bipartisan bill approved Gabriella Miller Kids First Research Act, authorizing $12.6 million per year for 10 years to support pediatric research at the NIH. And today, I'm happy to give you new developments. About a month ago in July, House passed the Kids First 2.0. We know still there are multiple steps for it to be signed into law, but nevertheless, I want to share with you this exciting news about the future of the Gabriella Miller program. The Kids First program starts with a basic hypothesis that there is an intersection between human development and childhood cancer. And through the many years of the program, our investigators, along with many researchers around the globe, are generating data substantiating that hypothesis. For instance, I want to point out, highlight validated clinical targets for oncology like BRAF, MAPK, and ALK mutations that are shared also with structural birth defects. More recently, a large study involved 10 million births associate birth defects with higher risk of developing cancer in the childhood as well as adulthood. At NIH, I work with a group of colleagues that are from multidisciplinary team from across the NIH with the leadership of the Common Fund and many other institutes forming a working body that oversees the program. At NIH, we call it a working group. So as part of the working group, I'm really excited to be part of this team to build a cloud-based genomic data resource to accelerate collaborative research, leading to better prevention, diagnosis, and treatment for patients and families affected by structural birth defects and pediatric cancer. What I learned is that Kids First program is empowered by the NIH EXO-1 mechanism, the Kids First sequencing centers, and Kids First data resources center. But always starts with the patient cohorts and your proposals. So proposals come to the EXO-1 mechanism, and I will explain a little bit more about that. Go through the NIH peer-reviewed system, and selected projects are then giving support, is an in-kind support for access to generation of data. So the NIH, through the EXO-1 mechanism, actively invest in data generation. And the EXO-1 investigator received that in-kind access to genomic sequencing that happens at the Kids First sequencing centers, the Broad Institute and Hudson Alpha. Once the sequence is done, the Data Resource Center compiled the genomic data with phenotypes and clinical data that investigators promised us in the beginning. And then the NIH approves the delivery of that data back to the EXO-1 investigators, who now have 6 months exclusivity to work with the data. After that 6 months, the data is released to the public through dbGaP and the Kids First portal. So this is a cycle that can also very often, and we have seen through the years, many of these projects generate hypotheses for new projects. In this way, the NIH fostering data generation, data sharing, and data analysis. But I will tell the data analysis is coming later, and I will tell you more about it, because the EXO-1 mechanism supports the data generation part. So I want to tell you a little bit more about the data delivery, which is an important step in this cycle, is this data cycle, if you will. Before delivery back, we go through an interactive and collaborative effort with our EXO-1 investigators to compile the genomic data with these phenotypes and clinical data. So it's a very collaborative process that we call clinical data review, and then it goes through a quality control before the NIH approve the delivery back to investigators. And as you can see in this graph, it's showing you that how projects being delivered to investigators since 2015. And as we won, most is blue, and some of the 2020 and 2021 are starting being delivered now. After 6 months exclusivity, the data then is released to the public for public access through dbGaP and the Kids First portal. And I would like to invite you all to register and look at the data in the portal. We already have genomes for almost 30,000 patients and families in our portal. And the good news is, even if you're not an EXO-1 investigator, you can have access to this data. This data is available for our NIH investigators, as well as wide community of researchers, both in the US and internationally. So again, I invite you to check it out, our data at the Kids First portal. The Kids First portal is powered by platforms such as Cavatica, and you're gonna listen more about it later today when you hear from the Data Resource Center. I wanna share with you recent data release. This is brand new. It's actually the first time the public sees this data releases that was from August to September, both in structural birth defects, 2 new studies that were released in the Kids First portal and now available for researchers to work with. And again, this is not only genomic, but also some transcriptome data that are associated with phenotypes and clinical data. And then also a new release on childhood cancers. So I invite you all to check the Kids First portal and look at the new data available. We expected to release multiple data sets in the next few months, both structural birth defects projects and new cancer projects as well, adding another 5,000 genomes representing patients and families. And I want to emphasize this is available not only for EXO-1 investigators, but also for the wide research community in the US and internationally. And this gives you an idea of the data access to the data we already have at the Kids First portal and through dbGaP. As you can see, since this data was released, not only the EXO-1 investigators access it, but it's increasing access through the time of all our data sets. Most of the data sets had also an increase from May to September. We can already see a significant increase in data access requests approved by the Kids First Data Access Committee. So we have a total of 56 projects, sequencing projects that the Kids First program funded through the sequencing centers, 30 publications, 27 studies available at dbGaP and the Kids First portal, with over 500 investigators already with access to this data. Are you going to be next? The Kids First program is not alone. We are part of a bigger ecosystem, data ecosystem that exists at NIH and outside NIH. I want to mention leadership of the Common Fund and Common Fund data ecosystem, our partnership with Include, and you're going to hear from today from our investigators working with Down syndrome. And also I want to mention our partnership with the NIH cloud-based platforms, interoperability, and CPI partners. We want to expand these partnerships, not only to create additional use cases and interoperability case, but also to start looking to functional studies on genetic variants that is being identified using the Kids First data. So our focus in 2023 is add additional data types. So genomics, proteomics, long read, diversity in cohorts. And you will hear in the near future some results from our proteomics and long read pilots. Enhance the data sharing and creating new collaborations. Again, collaborations to generate data, to analyze the data, to interoperate, name it, all are welcome, the more, the merrier. I want to finish the talk giving you a little bit of information about the EXO-1 mechanism. So what to expect, keep in mind, first of all, read and follow the guidance of program announcement. And the EXO-1 mechanism, check it out, the past opportunities, and the URL it's here will be available to you in this deck. But we also welcome that you reach out to us. So feel free to email me and I will direct you to our NIH staff that can answer your questions before you submit an application. So we expect to have new opportunities for fiscal year 2023. But as I said, look at the prior announcements so you can start preparing your proposals and reach out to us to discuss it if you have questions. Now, talking about data analysis, I want to talk a little bit, as I said, the EXO-1 mechanism, the main purpose is to generate the data. But then NIH also has support mechanisms, funding mechanisms for the data analysis. And I want to highlight the RO3 mechanism, the Kids First RO3, but as well as other institutions, RO3s, which can be used to analyze the data, not only analyze the EXO-1 data, but also even if you have your own data set and want to relate it to the data in the Kids First portal, that is also, we would welcome that type of applications. We also welcome additional adding phenotypes or longitudinal studies. So these are examples of proposals you can submit as a RO3 that will help you to analyze the data. And again, you don't need to be an EXO-1 investigator. As I said, these opportunities, both NIH EXO-1 and RO3s, are available to investigators in the US and internationally. As far as you're using Kids First data, we welcome you to be part of this community and collaborate. So I want to leave you with the Kids First vision, and I will welcome for questions. Thank you. Any questions? And if you don't have questions yet, but you like questions come into your mind, feel free to add to the Q&A box and we will be answering during the webinar. So I will proceed then. We're inviting our next next speaker.
Hi, Marcia, how are you doing?
Hi, Joaquin.
I am ready when you're ready.
Okay, I want to share here a little bit about you. So let me share my screen again and make. So Dr. Joaquin Espinosa is one of the Kids First Include investigators. He's being a champion for both programs. Dr. Espinosa is an executive director and professor in pharmacology at the Linda Clinic Institute for Down Syndrome at University of Colorado School of Medicine. When I asked him what about his work that relates to our webinar topic, he went right on the core of the question, which is work with multidimensional data sets in a transdisciplinary research team. So thank you, Dr. Joaquin, for joining us today. And I will stop sharing and then you can start sharing your presentation. Thank you again.
Thank you, Marcia, for the invitation to present on this topic. Definitely one of my favorite topics. Hopefully today, in the next 30 minutes, I will make a strong argument for the power of multidimensional data sets and collaborations to accelerate research, in this case, on one of my favorite topics, Down syndrome research. And hopefully implicit in that argument will be another argument for developing interoperability across various resources, not only within NIH, but also beyond. As you said, Marcia, I'm the director of the Linda Clinic Institute for Down syndrome, but I also serve as leader of the Include Data Coordinating Center Administrative and Outreach Core. And today, I would like to use the study of Down syndrome as a great example of the power of multidimensional data sets and collaborations. I like to say that Down syndrome is the ultimate challenge in precision personalized medicine. We've known for many decades now what is the genetic cause of Down syndrome. It's an extra copy of chromosome 21 discovered by Dr. Lujan back in 1959. Chromosome 21 was one of the first chromosomes to be fully sequenced, identifying 225 genes. There's no mutations, only an extra copy of these 225 genes. So then the question is, how we use this information to improve the lives of people with Down syndrome? This question is ever more important because for the first time in history, we have now a sizable population of adults with Down syndrome. People with Down syndrome have seen a massive elongation in their life expectancy. So it's the first time that we have millions of adults with Down syndrome that we can study. And what we have learned is that in addition to these core phenotypes of Down syndrome caused by the extra chromosome, people with Down syndrome are protected from many common medical conditions, for example, solid malignancies. Very rare to hear about an adult male with Down syndrome having prostate cancer, an adult female with Down syndrome having breast cancer. They also protected from atherosclerosis, hypertension, but unfortunately, on the other hand, they're highly predisposed to a number of conditions that also affect the typical population, such as Alzheimer's disease, several autoimmune disorders, leukemia, severe COVID-19. So clearly, if we study the impacts of the trisomy, we will be serving first and foremost people with Down syndrome, but also we will advance the knowledge of conditions that affect the general population. With that being said, it's also true that we cannot generalize about Down syndrome. There is massive inter-individual variability. All these individuals here are dealing with the extra copy of chromosome 21 in their own unique personal ways. So here plays 7 musical instruments in front of large audiences. Frank here at the bottom right gave an inspirational speech in Congress that triggered the Include project. However, other individuals in this slide may be nonverbal, affected by multiple co-occurring conditions, perhaps have a dual diagnosis of autism spectrum disorder. So clearly, we need a precision personalized medicine approach for the study of Down syndrome. So one key question is, what drives this remarkable phenotypic variation? Is it the genes on chromosome 21 or elsewhere? Is it epigenetic mechanisms? Could it be that the different phenotypes are matched to different endotypes, reflecting the transcriptome, the proteome, metabolome, microbiome, immune profiles? How about environment, lifestyle? socioeconomic status, cultural factors. So clearly, we need an orchestrated effort to test many, many possible mechanisms, hypotheses in one big. And that is where the NIH Include project comes. Include stands for Investigation of Co-occurring Conditions Across the Lifespan to Understand Down Syndrome, a phenomenal trans-NIH research initiative that started in 2018 and has already quadrupled the investment from NIH in Down syndrome research along 3 areas of focus listed there: high risk, high reward, basic science, assembly of a large cohort study across the lifespan, and inclusion of individuals with Down syndrome in clinical trials. In alignment with component 2 activities, here at the Resnick Institute at the University of Colorado, we are running one of the largest, most detailed cohort studies of individuals with Down syndrome, known as the Human Trisom Project. It's a panomics natural history study with a multidimensional biobank and a public research portal. More than 900 participants recruited since 2016, more than 600 of them with trisomy 21, the other 300 being controls. But we've been going at it with EXO-1 mechanisms, funding from the Global Down Syndrome Foundation and other mechanisms, generating tons of omics data sets, some of them listed here, and making those rapidly accessible through a number of means. So something that I think has been a place and an opportunity for innovation with the Human Trisom Project is in creating a very highly dimensional, multidimensional biobank, right? When a person with Down syndrome says, yes, I want to participate in a research study, that is a sacred moment. It's like they told you, yes, I want to marry you. You do not waste any biospecimens. If they give you blood, you treat that blood like liquid gold and fraction that blood into plasma, white blood cells, individual cell types, red blood cells, where they are giving you the blood. You may also ask them to, before they go, to give a urine sample where you can get live kidney cells to make iPSCs. You ask them to give you a saliva, stool, skin tape biopsy sample. And out of those samples, you can create many, many high content data sets that produce these highly multidimensional data sets for the study of Down syndrome. So if you do this right, in one study, you can answer what is the impact of trisomy 21 on the transcript, on the protein, the metabolome, immune cell repertoire, microbiome, global autoantibody profile, epigenome, functional genome. So here's a little message for you. Multidimensional biobanks lead to multidimensional data sets for collaborative discoveries of multidimensional impact. And that is what I would like to illustrate in the next 10 to 20 slides. Any cool results of this effort? Many. I want to tell you about one that we're very excited about. The first publication in this series of discoveries was a highly collaborative project where we look at cell lines and primary samples, and we did 3 high content data sets: transcriptome, proteome, short hair RNA screens. And in every data set, it was very clear that trisomy 21 consistently activates the interferon response. So what is the interferon response? It's part of the innate immune system. It's what we all use to fight off viruses. When a cell is infected by a virus, it releases interferons that bind to surface receptors in neighboring cells, leading to activation of an immune response. Now, too much interferon hyperactivity is not good. Among other things, it can create a risk of autoimmunity. There are 3 types of interferon signaling: type 1, 2, and 3, different receptors, different ligands. Yet they all transduce signals inside the cell through the JAK kinases. JAK1 is required for all 3 types of interferon signaling, and then you have TYK2 and JAK2. These kinases phosphorylate transcription factors that then in the nucleus trigger a transcriptional program inducing the expression of the so-called interferon-stimulated genes. Again, notice this will become important later that JAK1 is required for all interferon signaling. So more recently, we did a larger transcriptome analysis. This one is unpublished. 400 whole blood packed gene RNA tubes, 304 with trisomy 21 versus 96 control. And when you look at the volcano plot here, you see that on average, all chromosome 21 genes are elevated 1.5-fold, exactly what you would expect from having 3 copies instead of 2 of chromosome 21. But you can see in black hundreds of other genes that are consistently dysregulated in those with Down syndrome. When you get those genes and feed it to GSEA to ask what are the key signatures in that transcriptome, at the top of the charts, the interferon gamma response gene set. Number 3, interferon alpha response gene set. And then inflammatory response, TNF alpha signaling, many inflammatory signatures. The blood of a person with Down syndrome looks like it's fighting a viral infection, even though these individuals are healthy and there's no viral infection that we can detect. With those interferon gamma and interferon alpha inducible genes, we can create an interferon score that we can give to every participant. So you see here the interferon scores for the 96 controls versus the 304 individuals with Down syndrome. Interferon scores are greatly elevated in Down syndrome with a lot of inter-individual variability. Now on the side, we've been also running the COVID project, so we have access to identical samples from typical people without and with COVID. Look at that, the two plots are on the same scale. The transcriptome of the peripheral immune system of people with Down syndrome looks to a degree like the transcriptome of a typical person in the hospital battling COVID-19. Now, why would that be? Well, it so turns out that 4 out of the 6 interferon receptors are encoded on chromosome 21. So if you have trisomy 21, you have an extra copy of the 2 type 1 receptors, one each of the type 2 and type 3 receptors. In a short 200 kilobase piece of DNA, the 4 receptors are there. So you have an extra copy of it, you're likely to overexpress them, and that is correct. Here you see the whole blood transcriptome analysis, the 4 interferon receptors encoded on chromosome 21 significantly upregulated with a lot of inter-individual variability. Sure enough, when you ask them if the ISGs are elevated, yes, dozens of interferon-stimulated genes also significantly elevated in the bloodstream of people with Down syndrome. So then we set up a different collaboration to generate a different data set. This time we took the plasma samples out of the biobank and did a proteomics analysis with the SomaScan platform. Some of the key collaborators listed here. And the title of the paper says it all. Trisomy 21 causes changes in the circulating proteome indicative of chronic autoinflammation. Since then, we have repeated the experiment with 3 times samples. I'm showing you here plasma proteomics of 419 research participants, 316 with trisomy 21. Once again, we feed the results to GSEA, top of the charts, interferon gamma response, interferon alpha response, many other inflammatory signatures. So here are a couple of examples. TFF1 is a protein encoded by a gene on chromosome 21. Sure enough, is up 1.5-fold in the circulation of people with Down syndrome. SAA1 is not on chromosome 21. It's a canonical inflammatory marker, highly elevated in people with Down syndrome with a lot of inter-individual variation. To have a better assessment of this, we did quantitative targeted proteomics looking at dozens of inflammatory markers. Sure enough, the blood of people with Down syndrome shows highly elevated levels of many immune markers. I'm showing you here the general marker of inflammation, CRP, as an example. People with Down syndrome show much elevated levels of interferon inducible and also many other cytokines in circulation. Then we went back to the biobank, and now we took the white blood cells and did a mass cytometry, also known as cytoph experiment, a flow cytometry on steroids, where we can measure doses of immune cell types and also gauge their degree of signaling. The title of the paper also self-explanatory. This experiment revealed global immune remodeling with multi-lineage hypersensitivity to type 1 interferon Down syndrome, now collaborating with immunologists that can make sense of this high content data set. So here the computer is organizing this highly dimensional data set into clusters that belong to specific immune cell types. People with Down syndrome have dendritic cells, monocytes, T cells, B cells, all the cell types that we have. But then when you ask the computer to compare many samples with trisomy 21 versus those without and look for differences, you see the plot here on the left showing that basically everything is different. It is hard to find an aspect of the immune system that is not either violet or red. And you have to look at the cell subtype level. For example, the CD4 T cells in total are no different in Down syndrome, but if you look within them, you see that some subsets are depleted in trisomy 21. Some subsets of CD4 T cells are elevated in trisomy 21. So if you read this paper, you will see that we look at 100 different immune cell types. This is a brief summary of what we saw. Increasing Down syndrome is interferon receptor expression, interferon signaling, high levels of fibrocytes, inflammatory monocytes, activated natural killer cells, cytotoxic T cells, plasmablasts. But at the same time, we see depletion of stem cells, classical monocytes, naive cytotoxic T cells, immature B cells, and so on. As part of this experiment, we have one antibody to measure the surface expression of one of the receptors on chromosome 21, alpha receptor 1. And sure enough, when you look at 100 immune cell types, with or without the trisomy, basically all cell types that we looked at have elevated surface expression of the receptor. The stem cells, the monocytes, the dendritic cells, the ILCs, B cells, green, always stronger signals than gray. So what would happen if your immune system is expressing more of this receptor in all these immune cells? Of course, the immune system will be hypersensitive to interference stimulation. So as part of this experiment, we took a sample out of the biobank and stimulated it with interferon alpha ex vivo, and then we used CyTOF to measure now inside the cells for the phosphorylated transcription factors, STAT1, STAT4, STAT6. And in every immune cell type that we looked at, there were multiple STATs that were hyper phosphorylated when stimulated with interferon alpha. So this is how you and I in black do interferon signaling. It's quiet, we put interferon, we go up. In green, this is how people with Down syndrome do it. They start oftentimes from a higher basal and then they overshoot. They are super responders every time to interferon stimulation. This is true also for non-immune cells. This is an experiment that we have published earlier. These are skin fibroblasts, not immune cells. And you titrate in different amounts of interferon alpha, beta, gamma. You monitor expression of ISGs like MX1, ISG15. Cells with a trisomy start in a higher baseline and then super induce these ISGs. And this is likely to be happening then throughout the human body of individuals with Down syndrome. Then we went back to the biobank, got the same plasma samples that we have used for proteomics, and now put them through a mass spectrometer to do metabolomics. This required yet another collaboration with experts in metabolomics like Angelo D'Alessandro, James Costello. Title says it all. Trisomy 21 activates the kynurenine pathway via increased dosage of interferon receptors. So here's a volcano plot out of that paper. Metabolites upregulated and downregulated in Down syndrome at the top of the charts. The most significantly upregulated metabolite in Down syndrome, something called quinolinic acid, which happens to be a derivative of another metabolite also elevated in Down syndrome called kynurenine. That is the elevation of kynurenine and quinolinic acid. It so turns out these two metabolites are tryptophan catabolites that are produced only during conditions of inflammation. It's an interferon-inducible pathway. Interferon induces an ISG known as IDO1, which is the rate-limiting enzyme in the pathway. That's how you get quinolinic and quinolinic acid. And this caught our attention because quinolinic acid is known as the inescapable neurotoxin. It's a molecule that if it goes into your brain, it will bind to NMDA receptors and kill the neurons with those receptors. This is called excitatory toxicity. Memantine, a drug that is approved for Alzheimer's treatment, is an NMDR antagonist that protects from this toxicity. Circulating levels of quinolinic acid have been associated with lower cognition in older adults with Alzheimer's in the typical population. It's also known convulsant, and we know that individuals with Down syndrome are at high risk of epilepsy and seizure disorders. So this was something that we wanted to investigate further. As I mentioned, interferon induces IDO1. Is that true? That's elevating Down syndrome? Sure enough. Here the fibroblasts without and with the trisomy, you can see those with the trisomy super respond, make a lot more IDO1 when stimulated with interferon. If you look now at the transcriptome of white blood cells, IDO1 is elevated in living people with Down syndrome in the Denver area. And when you monitor the correlation between interferon-inducible cytokines like IP10 and the quinolinic-tryptophan ratio, sure enough, you see a positive correlation. The higher your interferon activity, the more dysregulation of the kynurenine pathway you observe. Could go on and on, but I want to pause here. I hope I convinced you by now that Down syndrome could be understood in good measure, not completely, in good measure, as an interferonopathy. So what is an interferonopathy? It's not something that we came up with. It's a group of genetic disorders characterized by consistent upregulation of the interferon response. There are many of these. These are mutations in your innate immune system, tricking the immune system into thinking there is a virus. So you produce a lot of interferons even if there isn't a virus, like Cardi-Gutierrez, Savi, Candle, many others. Now they're all different, yet they all share a core set of phenotypes that are also shared with Down syndrome: severe neurological dysfunction, severe developmental delay, hypomyelination, seizures, smaller cerebellum, so on and so forth. So that led to the obvious question: would then drugs that inhibit interferon signaling improve health outcomes in Down syndrome? To answer that question, we took advantage of the fact that there are many FDA-approved medicines that are known to inhibit the interferon response by targeting the JAK kinases, the so-called JAK inhibitors. There are many of them done by different companies with different brand names. They all target JAK1, which is, again, required for all three types of interferon signaling. Some of them target JAK2, JAK3. They have many approved indications. So then we ask, can JAK inhibitors normalize the immune system in Down syndrome? And if so, what could be the potential benefits? To answer that, we set up a clinical trial for JAK inhibition in Down syndrome funded through the INCLUDE project. We wanted participants in the trial to have, hopefully, a clinical benefit. So we knew that 25% of adults with Down syndrome are affected by one or more autoimmune skin conditions like atopic dermatitis, alopecia areata, vitiligo, psoriasis, hidradenitis suppurativa. So we focus our attention on those participants receiving treatment for 4 to 9 months with one JAK inhibitor, the one that has the most patients in the universe, known as tofacitinib gelsons. This is the study design. Individuals with Down syndrome, 12 to 50, phase 2, single arm, open label, everybody gets the medicine for 16 weeks, potentially an extension arm up to 24 weeks. They must have moderate to severe autoimmune skin conditions. And one, define the safety profile. Why, you may ask? These are FDA-approved drugs. Well, they were not tested in individuals with Down syndrome. So we want to redefine the safety profile specifically for people with Down syndrome. And 2, can we lower all those immune markers that are to the roof in Down syndrome? And 3, can we improve the skin and other autoimmune conditions? And 4, what if there are benefits that go beyond the skin, perhaps in cognition and quality of life? So I'm going to give you some top-level results. We analyzed the first 10 participants earlier this year, 0 serious adverse events. We look at the safety data, we're good to go. We're now 15 participants, we're going to go up to 40. All 6 participants that had alopecia areata, autoimmune hair loss, saw hair regrowth to different degrees. The 2 participants with atopic dermatitis were cured. The 1 participant with psoriasis was cured. 2 out of the 5 participants with the boils with hidradenitis suppurativa saw improvements. Now, more excitingly, we saw benefits going well beyond skin deep. All participants showed decreased inflammatory markers. All 7 that had an autoimmune attack to the thyroid gland, so decrease in the autoantibodies to the thyroid. And then very tantalizing, remember, we don't have a placebo control arm, so caution should be exercised. Very tantalizing. Improvements in 1 measure of spatial memory, visual motor function, and anxiety and depression scores. Let me show you some of the data. 1 image is worth 1,000 words. Young man from Texas flying with a family to Denver, see if we can address his alopecia areata, 17 years old. Hair is not growing. Cytotoxic CD8 T cells are attacking his hair follicle. Within 4 months of taking this JAK inhibitor, big redhead hairdo. He's been telling us he's known as Ed Sheeran among his friends. We didn't know why. Now we know why, because he has that amazing hair. He's another participant. He couldn't travel all the way from Vermont to Colorado, so we monitor him with a colleague in Vermont. Psoriatic arthritis. Look at those skin lesions. Also, you see there is a walker. Person cannot walk unassisted. Within a few months of treatment, psoriasis lesions are gone. This young man now is back on the stationary bike, which made me very, very happy. I told you about the autoimmune attack to the thyroid gland. We can monitor this with anti-TPO antibodies. We have 7 out of 10 participants having above normal levels of anti-TPO. All 7 of them saw a decrease in TPO levels. Here's 1 participant, the time course. Within a couple of weeks of going on the medicine, you see the autoimmune attack to the thyroid disease decreases. And when I talk about potential improvements in neurological function, this is what I'm talking about. We have 1 participant with Down syndrome regression disorder, which I will explain in a minute. At baseline, we were asking this young woman to take a pencil and follow the path of the car of the motorcycle without lifting the pen, without going outside the path. She could not understand the task. She was obsessed with coloring the path, took too long, lifted the pencil too many times, went outside the path many times. At week 16, she clearly understood what she needed to do. She drew in a single shot the path of the car of the motorcycle. So what is Down syndrome regression disorder? It's a rare but very devastating condition affecting young adolescents and young adults with Down syndrome, characterized by catatonia, mutism, depersonalization, loss of abilities to perform activities of daily living. These individuals crash for some unknown reason. Now, there's evidence that a subset of these cases could be associated with inflammation in the brain, which made us wonder, could it be that DSRD is an autoimmune condition similar perhaps to autoimmune encephalitis? So now, funded by the INCLUDE project, we're going to do another trial focused specifically on Down syndrome regression disorder. We will compare the efficacy and safety of 3 therapies that are commonly used, 2 immune therapies and 1 psychiatric medicine. And this is a large collaboration, yet another collaboration, this time with neuroimmunologists and psychiatrists. I should point out, obviously, the previous trial is a strong collaboration with a dermatologist. So coming back to the immune markers, I'm showing you here the same data I showed you before. The interference scores highly elevated in people with Down syndrome. Our clinical trial participants come to the trial with interference scores that are about average for Down syndrome, but when they go in the medicine, they drop more to a normal range. So we're not fully depressing the immune system, we're bringing down the immune system down to the range that you would see in the general population. He said, Joaquin, you haven't shown us any longitudinal data. Well, let me do it. We've been following one participant for many years who even before the trial was taking tofacitinib for her alopecia areata, and she will take the drug, get the hair grown, stop the drug, alopecia will come back. So she gave us a lot of samples when on and off the drug. So we can see longitudinal what happens to her immune system. When she is off the drug, her interference scores are about average for Down syndrome. When she is on the drug, her interference scores are average for a typical person. You see the various interference stimulated genes here coming from a high place to a mid-range every time she's on the drug. So the medicine normalizes interference response without an overt massive immune suppression. Hopefully, I made my case that multidimensional data sets can massively accelerate the translational pipeline from the first paper that I showed you to the first participant, 4 years. 4 years from a basic science experiment to a clinical trial. Now you may say, Joaquin, you don't have any animal data. We do. You can check the paper in bioRxiv. If we normalize the copy number of the interferon receptors in a mouse model of Down syndrome, expectedly perhaps we rescue the immune hypersensitivity, but very interestingly, we also decrease the rate of congenital heart disease, craniofacial abnormalities, and cognitive impairments. So in conclusion, interferon hyperactivity could cause many health issues in individuals with Down syndrome, such as autoimmune disorders, severe complications from lung infections, including COVID-19. This research is illuminating potential strategies to benefit people with Down syndrome through immune modulation, such as the JAK inhibitors. And it is our hope that immune modulation will have multidimensional benefits for this population. Now, coming back full circle to my first slide. Coming back to multidimensionality and interoperability. Where is all the data that I shall show you? It's all over the place. Genomes in LiviGap and Kids First, transcriptomes in GEO, proteome in proteome exchange, metabolome, mastocytome. It's a nightmare. As a PI who likes to collaborate and walk out of my comfort zone, then I had to keep track of our data across a number of repositories. So this is one of the missions of the INCLUDE Data Hub, to take care of this problem and make sure that we can find all the data in one place and facilitate cross-analysis of multidimensional data sets. So I'm very proud to be part of this very talented group of the INCLUDE Data Coordinating Center. You see the PIs here listed in alphabetical order, and we are starting to address this problem. Now all the HTTP data can be found in one place. I can go now to the INCLUDE Data Hub, select the HTTP, and I see my participants, my biospecimens, my data files. I can click on the biorepository and I see my plasma, my white blood cells, my red blood cells, so on and so forth. I can go into the data files and I see my transcriptomes, my genomes, so on and so forth. And of course, we are very mindful that not only we have to integrate within the INCLUDE Data Hub, but also integrate with other resources. So if we put Kids First at the center of the universe here, we want to make sure that it's fully compatible with the INCLUDE Data Hub, but also with other resources, and all those resources compatible with each other. This is the most scary slide because I'm always concerned that I'm going to forget somebody. I'm clearly the mouthpiece for a very large team that is very highly collaborative and innovative. Members of the Resnick Institute, the JAK-I trial, the DSRD team, the included CC team, many funding sources for various of these projects. And with that, I would be happy to take any questions and criticism if the time allows. I see that I'm at 28 minutes already. Thank you, Marcia, again for the invitation to present at this important workshop.
Thank you, Joaquin. This was an amazing talk. So I would like to see if there are any questions from the audience. So I have here a great talk. Is interferon activity in Down syndrome age dependent? How does increased interferon activity explain the large variation of Down syndrome?
Wonderful question. Number one, interferon scores are not age dependent, but there are some cytokines that I show you elevated in Down syndrome that are age dependent, right? And we are addressing that. So we are giving, just like we're giving everybody with Down syndrome an interferon score, we're giving everybody with Down syndrome an inflammaging score, that is age-associated inflammation. We're trying to understand what that does. Is interferon score, can it explain the variability? Yes, to a degree, not all of it. So we know now that participants with higher interferon scores are more likely to have autoimmune thyroid disease, more likely to have a history of congenital heart disease as well. But there are other co-occurring conditions that we cannot easily track to the degree of interferon signaling. I should point out, Marcia, that to do this right, we will need to have longitudinal information on interferon scores, that a single time point may not be good enough for us to map it to the clinical reality. We may need to have multiple time points, then coalesce all that information and map it to the clinical record. Great question.
And I will actually get a segue from the comment you made in terms of longitudinal data, because this is the type of data, for instance, our RO3 can fund. So out there, this type of data, using the opportunity of applying for RO3, I want to mention that this is a great example of how this mechanism could help on the data analysis and adding additional data. I want to ask you, what is, I think we don't have questions. If we still have a minute for additional questions, so go ahead. If you still haven't added your questions to the Q&A, feel free to do now. And in the meantime, I'm curious. Well, first, I love that you put kids first as the center of the universe. So thank you for that. But my question to you is for researchers out there that wants to start doing genomic research in the cloud, using a resource like the Kids First resource. What will be your advice? How they can, what is the first step they should do? What will be your advice for that?
Create a user account in Kids First and include Data Hub. Down syndrome data in the, or most of the Down syndrome data in Kids First is also in the Include Data Hub. And then they are similar but different, you know. So if you know how to learn one, the other one will seem familiar. And then also sign up for the Include newsletter. You do this through the Include website, includecc.org, where we're going to keep you updated of any new data releases that may be populating the Data Hub. Right now, we have 5 studies in the Include Data Hub. Next week, we're going to have another 2. You know, next March, we're going to have another 3. So just like Kids First has been growing over the years, so would the Include Data Hub. So my advice is sign up, become familiar with the data sets, reach out to the data generators, you know, the Human Trisom Project, Philip Lupo, Stephanie Sherman, ABCDS, DS Connect, to the various studies that are providing data to Include Data Hub, and find out how you can collaborate.
And for the young investigators out there, what do you have 30 seconds wisdom? Yeah. Message of wisdom for the young investigators out there.
Young investigators, it is a new age. When I did a graduate school or postdoc, I could do everything. From cleaning the glass plates to do electrophoresis to analyzing the data and writing the paper. The paper had 2 authors, myself and my mentor. Those days are gone. Now, chances are that you're going to be doing experiments that, first of all, you cannot do on your own. And second, you'll not be able to make sense of the data without collaboration and resources like these, resources that we talked about. So start your training with a collaborative mindset, trying to understand how can you place yourself in the key strategic position in a network of collaborators where you can be the intellectual leader of the collaboration to accelerate discoveries. The days of scientists as lone wolves, long gone.
Thank you, Joaquin, again. It's a great honor to be here with you today. And if there are any other questions, we will keep answering even after Joaquin leave us. And I will call our next speaker. And let me go back to my agenda. And it's, again, a great pleasure for me today to be hosting Kids First investigators talking about the collaborative research and how they are using data across multiple conditions to foster new discoveries. And it's a pleasure to call Dr. Sobreira to the podium. Are you there?
I'm here. Can you hear me?
Yes. Hi, Nara. Nice
Hi,
seeing
Marcia.
you. Yeah,
Nice to
and
see you.
Nara and I, we share something in common. We are both from Brazil.
Yes, and I'm here now. I'm actually in Brazil today.
Oh, okay. So that's great. So Nara is joining from Brazil. And that, again, is an example of our, like, how our collaborations go far beyond borders. So I asked Nara to share how she would hope to impact the audience today. And so she wants to highlight the importance of genomic research and also walking through multidisciplinary teams. So Nara, I will let you tell your story. And thank you again for participating here today.
Thank you, Marcia, for having me today. So let me share my screen with you all. Yeah. So I hope you can see my screen now. I'm going to talk to you today about our work studying enchondromatosis. We have done whole genome sequencing to understand better these diseases, and I will tell you a little bit about our results so far. So enchondromatosis is a group of diseases characterized by enchondromas that are benign cartilage tumors. So the patients have multiple cartilage tumors. And there are few different diseases where that happen. And some of them are listed here. They are mostly very rare. And for some of them, we know the gene, and the gene are mutated as a germline, and they follow a Mendelian mode of inheritance, like this is spondyloenchondromatosis, that's autosomal dominant disease caused by mutations in COL2A1, and the spondyloenchondrodysplasia with immune dysregulation, autosomal recessive, caused by mutations in ACP5, and metachondromatosis, that's actually how I got involved with these diseases when I identified that was autosomal dominant disease caused by loss of function mutations in PTPN11 during my PhD back in 2010. There are some others that we don't really know the cause, the genetic etiology, like the metaphyseal enchondromatosis with V2-hydroxylglutaric aciduria and the genochondromatosis. But the two most common ones, they are Olea disease and Maffucci syndrome. So Olea disease, the patients have mainly the enchondromas, and Maffucci syndrome, the patients have the enchondromas plus vascular anomalies. Vascular anomalies type overgrowth, like what we see with the PIK3CA diseases, for example. In both of these diseases, and they are the most common of the enchondromatoses, and in both of them, we do see a prevalence of 30% of these patients having chondrosarcomas. So there is a transformation of the enchondromas to chondrosarcomas in 30% of these patients. Some of these patients have the enchondromas and or the vascular anomalies as soon as they are born. And the chondrosarcomas, they appear later, a little bit later in life. And these enchondromas, independent of the chondrosarcomas and the vascular anomalies, they are very, very harmful to the patients. They can cause severe deformities, with leg length discrepancy, arm length discrepancies, and these overgrowth that you see of the vascular anomalies, they can even cause amputation of fingers, toes, hands, feet, and even legs and arms. So it is a very debilitating disease that affects the patients from childhood. And then later, they will go on, many of them, to have malignancies like chondrosarcoma that I just said, but also few others. So 30% of the patients go on to have chondrosarcomas, but up to 50% of the patients go on to have a malignancy of some kind. Most of the malignancies that are affecting these patients after chondrosarcoma, they are the brain, the gliomas. So up to 10 to 15% of the patients with early disease go on to have gliomas, and a little less of the patients with Maffucci also go on to have gliomas, like 5% of them, between 3.8 and 5%. A few other patients also have other kind of malignancies, like they affect the ovaries in some of these patients, up to 5% in both early disease and Maffucci syndrome. And in Maffucci syndrome, many of them, about 5% of them, end up having malignancies of the vascular overgrowth. So in general, in both cases, up to 50% of the patients end up with a malignancy of some kind during their life. So this is a disease that puts together the congenital anomalies. Many of these patients are already born with the deformities, the skeletal deformities caused by the enchondromas, or the vascular anomalies, and cancer that can happen during the childhood, but it's actually more common a little later in life. So these diseases are mostly isolated. There is no case that is known of early disease and Maffucci syndrome that is actually familial. As far as we know, all the cases that have been described and all the cases that we have actually enrolled in this project are isolated cases in the family. And there is, when I started investigating this disease back in 2010, nobody knew anything about them. Like there was no gene whatsoever that was associated with them. But then in 2011, two different groups identified in 80% of the patients mutations in IDH1 or IDH2 in the samples from enchondromas, chondrosarcoma, or vascular anomalies of these patients. These are the gain of function variants in IDH1 and IDH2 that we see in the patients with many other cancers, mainly like gliomas and leukemias. And if you are familiar with these mutations and in these other cancers, you know that usually in gliomas and leukemias, IDH1 and IDH2, they do co-occur with other variants in other genes. So when we found out that these variants were present from these other two groups in the patients with early disease and Maffucci syndrome, our first question was, what's the role of these genes in these patients? And are they the only gene in these patients that are causing the patients to have the enchondromas? But how also could some of the patients go on to have the chondrosarcomas and the other malignancies, and some do not, while 80% of them have the IDH1 and IDH2 independent of having malignancies or not. So we decided that we wanted to investigate better and investigate not only the tumor of the patients and not only IDH1 and IDH2 mutations specifically, but do an investigation that was genome-wide, germline, and tumor of these patients to understand better what was going on in the mutational landscape, and with that, maybe get to a pharmacological treatment that would avoid these tumors to develop from the beginning. So our ultimate goal is actually to try to find a medication that would stop the enchondromas to grow as soon as the first ones appear, or even make the ones that are already there to decrease in size and avoid all the skeletal deformities that these patients develop that are really very debilitating for them. So our hypothesis is that these syndromes are cancer predisposition syndromes, and that they are caused by germline variants or early post-zygotic variants that in combination with mutations that appear later will cause not only the enchondroma progression, but also the transformation of these enchondromas and chondrosarcomas and the appearance of other malignancies. So we are actually looking for multiple genes in each of these patients that would act together to cause the disease the way it is appearing in the patients. So to do that, we decided we needed to sequence a large number of patients. And the Kids First project was a perfect opportunity when I had just started as a faculty at Hopkins to actually do that. But at that point, we didn't have all the patients that we needed. And we actually, after we got the grant, we went on to find these patients. So here it was the first time as a faculty that I really had to collaborate, but not only with other physicians, right? I started by reaching out to surgeons, dermatologists, to everybody who would maybe be seeing patients with these disorders to find out who could send us samples for the project. But turns out these are rare diseases. So there's no one person that sees dozens of these patients in a year. And the best collaboration that I actually set up was with the patients themselves. When I realized that it would be very difficult to me to find all the physicians that together we could put the samples all together for the project, I decided to reach out directly to the patients. And that was one of the best collaborations that I did during the beginning of my career and has been on up to now, thankfully, was with the patients themselves. So I wrote a protocol, I submitted to our IRB at Hopkins, and I was allowed to reach out to these patients directly through the media platforms. And they are the ones that actually made this project happen. So they really collaborated with us on connecting to each other and informing each other of the project and getting patients to reach out to us directly to show their interest and participate with us on the project. So as of today, we have actually sequenced about 130 patients, but I'm going to show you the data of 94 that we have analyzed so far. So we performed whole exome sequencing or whole genome sequencing on 94 patients. We analyzed all 94 patients to date, and 71 of them have Ollier disease and 23 of them have Maffucci syndrome. And of these 94, we have samples from 68 trios. And these patients were sequenced through the Kids First, but also through the Baylor Hopkins Center for Mendelian Genomics. So the genomes were done through the Kids First, and the exons were done through Baylor Hopkins. And the exons that are done through the Baylor Hopkins are mostly the ones that we didn't have trios because the Kids First project was intended for the trios specifically. So what have we found with our initial analysis? We identified variants that are rare with a minor allele frequency lower than 1% encoding in 6 genes: HIF1 alpha, VHL, IDH1, IDH2, KDM4C, and CDKN2A in about 22% of our patients. So it's like 21 out of the 94, and 14 of them have Ollier disease and 7 of them have Maffucci syndrome. So why is it important? Why these genes specifically? Well, what we noticed when we were doing the analysis and we are finding the mutations repeating on these genes is that these genes actually, they all belong to the HIF1 alpha pathway. And that was striking to us and made us focus a little bit more on this pathway in these patients. So the one question that we had to begin with before we could really get into functional studies for these genes was, is it just by chance, right, that we are finding these variants in these genes among our patients? And these are germline variants. So we are focusing here today on the analysis that we are doing on the germline sequencing. And we performed this burden analysis. We had let me just, oh, yeah. So we had about 2,100 samples that we used as controls. And then we had the 94 samples that were the group of patients. And we compared to look in both groups, what were the number of variants that were rare coding, not including the synonyms, among the patients compared to the controls, to see if the rate of variants among the patients in these genes was higher than among the controls. And we identified that the variants in HIF1 alpha, in VHL, and in IDH1, the germline variants, were actually more common among the patients than in controls, with a difference that was statistically significant. And you can see here the corrected p-value of this comparison. So that made us really be more excited about the pathway, about the variants, about the genes, and want to understand more how this pathway is affected in these patients and how actually does it interact, like how does it relate with what we already knew about the IDH1 and IDH2 mutations in these patients. So this is just an overview of the HIF1 alpha pathway. You can see here on normoxia, we have usually degradation of HIF1 alpha by a complex that is formed together with VHL. And if we have hypoxia, this complex doesn't form, HIF1 alpha is not degraded and goes on to activate many pathways downstream. So you have here the pathways like angiogenesis, erythropoiesis, metabolism, cell survival. These are all pathways that are downstream HIF1 alpha that are activated at hypoxia when HIF1 alpha is upregulated. So, and in this, here you can see that we found mutations in HIF1 alpha itself. It's actually the first time that mutations in HIF1 alpha have been identified in patients and related to a disease. It has never been described before, mutations in HIF1 alpha itself. Then we have mutations in VHL that we all know cause VHL syndrome. Our patients do not have any features of VHL syndrome. They do have vascular anomalies, and some of the patients with VHL syndrome have vascular anomalies, but a slightly different kind of vascular anomalies. They are not exactly what we see in the Maffucci syndrome. And also the malignancies in the VHL syndrome are different of the malignancies that we are seeing in the patients with Maffucci or earlier disease. But our patients have mutation in VHL, and you see here, we have actually 6 patients with mutation in VHL, 7 patients with mutation in HIF1 alpha, and 3 patients that had germline mutations in IDH1. One of them was a gain of function mutation that we already know, the one that we already know, the R132, but the other 2 were not the gain of function that are well known and described in the cancers. So here we also have mutation in CDKN2A. That's another inhibitor of the complex formation that has to happen during the hypoxia. So HIF1 alpha is actually activated. And we have KDM4C that is also a co-activator of HIF1 alpha during hypoxia. So we have these genes here, HIF1 alpha, VHL, CDKN2A, and KDM4C with mutations. We do have also 2 patients that have EGLN1 mutations, but because the number of patients was too low, we are still gathering more evidence to actually, before we actually go on and study more EGLN1, sorry. We didn't find mutations in EGLN2 or EGLN3 that are all here acting in that step of the pathway during normoxia. So how this pathway actually correlates with IDH1 and IDH2 that we already knew are mutated in patients with early disease and Maffucci syndrome. The mutations in IDH1 and IDH2, they are gain of function because they actually create a new function for IDH1 and IDH2. What happens now is that this alpha ketoglutarate, that's the product that would be created by the normal regular IDH1 and IDH2, is not created anymore. What is formed now is D2-hydroxyglutaric acid, sorry. And these D2-hydroxyglutaric acids are oncometabolite, and it actually acts by inhibiting some of the functions of these, inhibiting KDM4C and upregulating EGLNs. And the upregulation of EGLN would actually cause downregulation of HIF1 alpha. So this is how the HIF1 alpha pathway is connected to IDH1 and IDH2 mutations. So we really got very interested on that because we are finding more mutations on these genes that are in the HIF1 alpha pathway. That is a pathway that is connected, directly connected with IDH1 and IDH2. So, as I told you, these patients also have cancers like gliomas, and IDH1 and IDH2 are also very well studied in the setting of isolated gliomas. And also in the chondrosarcomas, 50% of the patients with isolated chondrosarcomas have the same gain of function mutations in IDH1 and IDH2. So now that we were finding the mutations in the HIF1 alpha pathway in our patients, our next question was, what about patients with isolated gliomas and isolated chondrosarcomas? What about this pathway in these patients? Are these genes mutated in these patients also? So that is actually the title of this lecture, is actually the title of our RO3. So we submitted the RO3 that Marcia mentioned a few times today. And we wanted to do the analysis of this data, but actually gathering the samples, the information on samples from gliomas and chondrosarcomas to study the genes that we find in the patients with Olin-Maffucci, independent of HIF1 alpha or not in these cohorts, but also do one analysis that's focusing on the HIF1 alpha pathway. So that started us on the path of trying to track down where to find the data and how to have access to the data. And I have to say, the speaker, Joaquin, just before me, just talked about the nightmare that is to know where all the data is. It is very complicated to find where all the data is. Like, where can I find sequencing from patients with glioma? Where can I find sequencing from patients with chondrosarcoma or other tumors that are related, like osteosarcoma, for example? So it's very easy to go to the Kids First database and find what is there, but it's not easy to know what else is out there in other databases. So that would be a very good thing. It would be to kind of centralize or at least have a place where we go and list all the places where these diseases may be, the samples related to this disease may be, because we end up having to go everywhere to try to find it, and not always we find. I'm sure sometimes we miss because we don't know, we didn't look at the one or other place that could be. So we actually found that in the Kids First website, we found that there were 816 patients from the Pediatric Brain Tumor Atlas that had been sequenced, and these patients had gliomas, and that we could access the data through the Cavarica and through the Kids First program, DRC and Cavarica, and we did that. Then we started looking for samples that are related to chondrosarcoma or sarcoma, osteosarcomas, and there are 383 patients that are part of osseous and chondromatous neoplasm from the Target OS project. That one, I still didn't figure out how to access the data. So I'm actually working with some colleagues from NIH and NCI to try to figure out how to find the data. And this is one of the data that we are trying to figure out how to access. But also through the Kids First, I found that there are some osteosarcoma projects and Ewing sarcoma projects that are already are available to be accessed, and I'm working on accessing them. And I just got through dbGaP access to whole genome sequence of 13 patients with osteosarcoma from the Osteosarcoma Genomics Project. So first of all, these are not samples that are highly sequenced. Many samples are sequenced, like few samples of patients with this disease are sequenced, and they are a little bit everywhere. So it's a little hard to figure out where to go to find all of them. Doesn't look like you are going to go to one place and get all the samples. You really have to go to multiple places to try to get as many samples as possible. But we are working on that. We already have the glioma samples. We already have some osteosarcoma samples, and now we are trying to get the chondrosarcoma samples and the osteosarcoma and the Ewing sarcoma samples. Here is the samples that we got from the patients with gliomas from the Pediatric Brain Tumor Atlas database through the Kids First Cavarica. And we got the VCF files from these about 800 something patients, and we already harmonized the files. They're all harmonized through the Cavarica pipeline to the same way our samples are harmonized in Cavarica for the Oliver Mafucci project. And now we can start the comparison. So our comparison will first be patients versus controls, our patients with Oliver Mafucci versus controls, and patients with gliomas versus controls. So we are working on that. And another step that we had to overcome was to find the samples that we would use as controls. And we figured that out. Some of the samples are going to be used as the samples that we sequenced through the Baylor Hopkins project. And some of the samples that we are going to use as controls are the samples that we sequence as part of the Kids First, because I actually collaborate with two other projects that were sequenced through the Kids First, the bladder extrophy and the face syndrome. And we are going to use each other's data as controls to do this burden analysis to find out what of the 20,000 genes are more mutated among the patients compared to the control. We are starting with the germline analysis, but we are also going to do the tumor analysis for the samples that we have that were sequenced as part of tumor projects. So I think that's where we are right now. And with that, I wanted to, of course, thank everybody who has been working with us on this project, different PIs in different labs, in different hospitals, the patients, and also the NIH NCPI team that has helped us a lot with finding the samples, getting access to the samples, and with the whole understanding the process of how to access everything through Cavarica. And it has been actually very helpful to have everything in Cavarica and be able to harmonize the entire pipeline through the Cavarica pipeline to do the analysis that we are doing now. So thank you very much, Marcia, for the opportunity. I'm happy to answer any questions that you have.
Thank you, Nara. And this is a great example, again, how we're collaborating. And this is a collaboration with the NCI and Kids First. So thank you for bringing this study. And because of the time, I will ask for questions being, you can add your questions to the chat or the question box, and we will answer. But I will ask that we move to the next speaker so we can give time to everybody. But again, thank you, Nara, so much for joining from Brazil and sharing your research and this amazing collaboration. We will be looking forward to hearing more from you after you can get all the studies together.
Thank you very much, Marcia.
And with that, I will invite Dr. Hakon to join us here. And let me share here a little introduction about Dr. Hakon, who is the director of the Center for Applied Genomics at the University of Pennsylvania School of Medicine. Dr. Hakon, are you still there?
Yes, can you hear me?
Yes. Hello.
Yeah.
So with that, I will quickly pass the word to you because I know we have such a short time, but we are eager to hear your presentation. So thank you so much for joining our webinar.
And can you see my slides and you can hear me?
Yes, we can hear you fine.
Okay, great. Yeah, so I'm going to tell you about an extra one program that we have at Children's Hospital Philadelphia. This is actually quite a unique cohort because we have several hundred children who have both congenital malformations and have suffered from a pediatric cancer, and in one and the same child. And the question is, do they have a single gene defect causing both, or are these two independent factors? And so we are looking sort of at the correlations between these sort of two phenotypes with respect to the genetic underpinnings. Now, as you know, if you have multiple, or if you have a congenital anomaly or malformation, you are predisposed to have cancer. So there's perhaps sort of reasons to believe that there may be overlapping pathways or genetic variants that contribute to both of those. This project is sort of set to look into that. And here is the cohort selection, and I am not going to go over this definitions here in detail, but these are the electronic medical record codes from the ICD-9 and 10 codes that we use to capture congenital anomalies and also to capture cancer. And there were 1,464 probands that we identified, and as you can see on the slide, 832, they overlap between cancer and having a birth defect. Now we sort of, had envisioned that this would be a larger overlap, but a number of these individuals had sort of, overgrowth syndrome and other benign growth parameters. So we removed them, and they are sort of, listed just as birth defects. And a few of these individuals had cancer alone without a birth defect, but the vast majority had both. And there were 141 trios, so we could sort of look at the parents as well. And this is a slide that just shows the age distributions in males and females, and so you can see that the average age is probably around 18 or 19 now, but these are pediatric patients when they were recruited. And then you can see the ethnic background and the race split up between males and females. Now this is sort of dominantly European ancestry individuals, but there are 157 African-American males in the study and 123 African-American females. And then you see the family structure, as I said, and the broken up trios between males and females. And this is sort of, that most people are aware of. I mean, the samples whole genome sequencing came from the Broad, one of the sequencing center, and we sort of, drove the phenotypes generating HBO term. And this was sort of, scored by Sortan and AWS Comprehend Medical. There was comprehensive analysis for single nucleotide variants, as well as copy number variants. And we used programs that we developed largely at our center at the Applied Genomics at CHOP. One is called GD Cross, and this is a variant sort of, ranking and prioritization software that sort of gives you pathogenicity and the most likely variant based on the input of the HPO terms. And the copy number variants are analyzed by the PennCNV and sort of the internal programs we have built at CAC. All sort of, chromosomal structural variants were visualized for validation, and we looked into similarly mitochondrial sequence analysis, fusion, expansion defects, and so on and so forth. And this sort of just shows the pipeline. There were 1,806 individual totals, other individuals, family members to the 1,400 probands. And you can just sort of see there were two steps really that we, for the data curation and sort of all kinds of tool development and benchmarking that we needed to do. So this GD cross, as I said, really is a powerful tool to pull out variants and rank them with respect to causality. And we could actually validate several of these variants because some of these kids, you know, had actually also clinical molecular diagnosis in the alphanumerical record, although vast majority didn't, but we could sort of randomly validate some of them. And then the downstream analysis and functional validation sort of followed, and this sort of discovery pipeline shows that, you know, we looked at both coding and non-coding region, the burden analysis, and the HPO clustering was sort of critically important for the for the downstream sort of, you know, function of the GD cross program. And then we, you know, when we came up with variants known or novel, we looked into gene matcher for, you know, there were other cases reported, and we took other variants into functional analysis if they were novel to establish and validate their sort of causality with respect to these phenotypes. And here you can see sort of a summary of the phenotypes in both in the cancer group and in the HPO terms. And so you can see the type of cancers that these individuals had, most commonly in the central nervous system. Just because of time, I'm not going to go into this in detail, but you can see that, you know, essentially every organ system was involved in terms of that there was a wide variety of cancer types, with nervous system cancers, neurofibromas, and leukemias being the most common. And then the HPO terms in terms of organ affected based on the phenotype input. Similarly, nervous system probably was the most common one, 682, and cardiovascular, musculoskeletal, digestive system, and essentially every organ system was inflicted in some individuals. And here's a summary of the molecular findings. There was actually, you know, a sizable number of individuals with chromosomal sort of, you know, significant chromosomal anomalies, aneuploidy, mostly trisomy, and we heard a talk on that here before. There were, you know, few monosomy and Turner syndromes, and then you can see here sort of the type of mutations that were, you know, for the homozygous or hemizygous that accounted for 17%. Frame shift, nonsense, splice sites, and other in-frame variants. Missense was 184 that have been uncovered for today. And then you can see the copy number variation analysis we've done, and the size ranges in terms of the, you know, we've captured copy number variants that are both small as well as large, apart from the whole chromosomal aberrations. Pathway analysis was performed similarly across the birth defects and birth defects and cancer. And this, of course, is always helpful to understand the overlap and the integration of the of these phenotypes in this sort of venue. And anyone can look at the slides and look at this more carefully afterwards. This is the sort of standard pathway analysis that we performed. One of the things that we were interested in knowing, as I said, was to see if there was a single molecular diagnosis for both the congenital anomaly observed and the cancer. We, you know, have multiple such examples, such as the MNX1 here that causes sacrococcal teratoma, and also anorectal malformations. There's the Wilms tumor gene with nephroblastoma and various sort of, you know, urogenital malformations. And there's the porcine gene, focal dermal hypoplasia, so laryngeal papilloma and actually other sort of one signaling related cancers have been described from this gene, as well as congenital heart defects and microcephaly. And so these are just sort of representative example of observation where the same gene is responsible for both the cancer and the congenital malformation. More often we saw the opposite. There were dual molecular diagnoses. For example, the DICER3 gene, pleural pulmonary blastoma, and TDF-B2, the Lloyd-Dietz syndrome, developmental delay, mitral regurg, and alike, and the other one. So this individual had clear distinction between these two types of phenotypes, and this is just sort of scaling that a bit. This is not showing nearly all of the dual findings that we have, but just giving you representative sort of and showing where they are located in terms of the tissue type, the SCN1A, obviously epilepsy gene, and neurofibroma gene causes neurofibromatosis, obviously a tumor that can turn into malignancy. ANCAR-D11 is a gene that causes the KBG syndrome, whereas the TSHC1 is known to cause colon cancer. You have 14q12, which causes Rett syndrome, or deletion in that region causes Rett syndrome. And the PROCAR2 is a gene that causes Kalman syndrome, hypogonadalism, and sort of anomalies around that axis. And MYH6, well-known cardiomyopathy gene, LEMOX-T3, a gene known to cause cancer similarly. And you have the exit, the exocytosis, which also, turns into hereditary conditions and cancers. Well-known cancer genes, KMT, 2D, and certainly P10 and alike. So I'm not going to count through this in every single gene, but you can see that that there are sort of independent pathways in a number of these individuals that are separate causes, but there are also a large number of individuals who have a single cause, which is very interesting. We similarly looked at copy number and structural variations. This just to sort of show the type of size range of deletions and duplications that we can capture. I guess those slides represent the slides here. They show deletions. There's a 12.8 multi-gene deletion here shown, and you see a 10 kb multi-axon deletion here with the center involved. You have loss of heterozygosity, as you can tell, and there's only one allele present, either the A or the B allele in this region. And then single axon deletion, we can get down to that granularity with the whole genome data very easily. And obviously, a single axon defect can be sufficient to cause a phenotype. And here's just sort of, this is actually plotting 22q11.2 deletions and duplications. And you can see the signature patterns here. And this is sort of really, allows us to train algorithms almost in an AI type fashions to sort of, screen through the entire data set to come up with all deletions that are, segmented into AB, AD, AC, etc. And you can see a clear evidence of deletions or duplications were specified for 22q. So these, whole genome sequence data are very powerful for this type of analysis. So I wasn't going to go further into this, but this is a very unique data set. As you can tell, HPO terms are really the phenotypic driver, and then the input is into the GD cross, where we sort of, capture variants, annotate those variants, prioritize them with respect to disease risk for causality, The EMR phenotyping, obviously, is critically important, and you can do all kinds of burden analysis and cluster analysis. And I sort of, wasn't going to go into that for the sort of this short presentation. But we have, very accurate copy number variation analysis where we have causal genes captured by PenCNV and sort of, using a program called ParCNV2, which Joe Glessner in our group drove and is currently in press. So, this type of a program just sort of, shows you the capacity of working within the Kids First program. And obviously these actual ones that are funded through this mechanism are highly enabling, and obviously opportunities to subsequently integrate and work together with other groups to expand and scale and grow these type of discoveries that we have. And this is the team that sort of, has worked on this internally. Deborah Watson, Amir, and Shang, sort of with Joe Glessner leading the effort. And obviously want to thank the NIH and the Kids First program and everyone there for their support for enabling this. So with that, I'll stop. Hopefully we captured a couple of minutes, but we'll take it from there.
Thank you, Dr. Hakon. It's a pleasure host you. And so let's see if there are any questions here for Dr. Hakon. You can also feel free to raise your hand. But as we are tight on time, I will move on with the program. But I want to kind of summarize here what we saw so far. Basically, Dr. Espinosa telling us how a multiplicidade of data and also multidisciplinary teams can accelerate discoveries to get closer to patients. And then Dr. Sobreira with highlighting that doing genomic research in the cloud is not only about data access, but finding the data, finding cohorts and Kids First data resources, a focus on make it easier for researchers to find the data and combine data sets. And Dr. Hakon, thank you also for highlighting how analyzing the data, especially for some of these data sets that has a diversity of phenotypes, is also a challenge. And again, the Data Resource Center is trying to help researchers with tools to prioritize the genetic variants and phenotypes. So thank you for all of you to share your experiences and how you collaborate and how you're using the data from the Kids First Data Resource Center. And now we're going to hear from Adam Resnick. Many of you may know him as he is the director of the Kids First Data Resource Center. And I will not take more of his time and pass the word to him. Adam.
Thanks so much, Marcia, and others. For the sake of time, I'll actually be speaking on behalf of those who have done much of the work, and I really want to commend Yaron Guo and Jeremy Costanza, from the Kids First DRC side for developing resources that really take sort of an orthogonal approach to some of what you heard already today from Nara, for example, supporting the queryability and findability of the resources investigators might want to access by leveraging gene-centric queries. And one of the first challenges that investigators often encounter, and we in the DRC continuously try to support and improve how we describe and define the resources within the program, is what data can I access right now or query, and what data do I have to approach dbGaP and requests. And this has been especially challenging for investigators as the program has grown, and we have tens and tens of different projects that each one of them has their own dbGaP requisition. And it's further complicated by the fact that there's differential access layers within Kids First, depending on the sensitivity of the data. So cancer data sets and their or other data sets that have expression counts or gene fusion calls are within a register tier that does not require access from dbGaP. And likewise, tumor normal studies don't require dbGaP provision access, but source data or variant data often does require dbGaP accessing. And it's that paradigm of complex layers of both access management and governance, aligning with NIH best practices and policies, plus the queryability of those resources for investigators that the DRC continues to try and improve and solve. So I wanted to just toggle back and forth between presentation, and I'll try and make it short, and the portal, just to give you sort of a real-time view of that process. So investigators essentially, by accepting the terms and conditions of the portal, immediately enter the portal into the registered tier framework. And within this framework, the system already automatically knows and understands which data sets you already have authorization for within the portal from dbGaP. But as we've highlighted before, and I won't go through today, the portal does enable users to explore the cohort based on phenotypic and other variables, HPO terminologies, to create cross-disease cohorts. And for example, Nara and Hakon's cohorts are available for further exploration. That's one of the really exciting things about the Kids First program, is that data is released in very rapid order. So, all the work that Hakon just described is based off the data that's currently available within Kids First for you and others to explore, both phenotypically and molecularly. Similarly, the brain tumor data sets that Nara described are also available within this portal and can be, again, accessed, and likewise, Nara's own EXO-1 cohort for exploration. The challenge investigators often face is, how do I know which data sets to explore or utilize? And, I just wanted to highlight the differences between cancer and non-cancer cohorts, for some of that exploration. So here I just chose that same brain tumor cohort that Nara mentioned that she gained access to. And cancer data sets essentially permit users to explore the molecular data for the tumor normal setting. So here I'm just clicking at a single patient within that cohort, and we've empowered the PedsBio portal environment where users can gain row level data for this one patient and see all the derived mutational data for this particular patient. You can explore entire cohorts within PedsBio portal, and you can immediately query genes of interest like IDH1, for example, or IDH, as Nara mentioned, and explore those mutational settings within the context of the assembled data. So here I chose, IDH1 and IDH2, and I can select these samples, and then I can actually review the specific mutations for that cohort within this setting. And the system will allow us to see a combination of the actual specific mutations across the genomic landscape of all of these tumors. Now, this study actually combines data sets across multiple data sets that we have been harmonizing, and I'll touch on that briefly a little bit later, really in alignment with Nara's comments. So here, for example, you can see, a cross-cancer cohort that includes IDH1 and IDH2 mutations. But the challenge that investigators often face really relate into the cohorts for variant discovery. And when we actually began doing the analysis on behalf of the DRC, it was clear that this was going to be a big challenge. And specifically, it's a challenge because there is a very large number of variants. And even actually, if you review simply the known pathogenic and annotated variants, there are still a lot of variants. So here is actually the known space of currently released data sets where we're just looking at the number of ClinVar annotated pathogenic variants across all the cohorts. Here at the beginning is Hopkins cohort with the known annotated pathogenic variants. But known variants are typically not the challenge that investigators face. It's typically the unknown variants that are the challenge to phenotypically align. Let me just move my Zoom. So specifically, hold on. There we go. So specifically, we developed a variant explorer framework that allow users, even in the absence of dbGaP accession provisioning, to explore the context of the mutational setting for kids' first data sets. So here, for example, you can begin by looking at pathogenicity, and I'm just going to essentially highlight all the annotations, and I can already show you the data that came supported that chart that I just displayed, showing that there are more than 4,000 and already annotated pathogenic variable. And this table here essentially highlights the individual variants and the studies and the number of participants that are there. So for example, I can click on this particular number here, 3, and it'll essentially, what it'll do is it won't show you the row level data for that particular patients, but it'll show you the 3 studies that essentially contain that variant within them, allowing you as an investigator to further curate those studies phenotypically through the cohort builder, which we've demonstrated in the past, but then assess whether or not you would like to further request it. The other thing that I wanted to highlight briefly before the end is that many investigators already have information about a gene of interest. You heard this from Nara, for example. So Nara mentioned, for example, IDH1 or IDH2 mutations. So I'm just going to put IDH2 as a use case here. And immediately you can see a list of variants at IDH2. There are more than 2,600, but then you can further filter those variants and review them. So here, if I click on ClinVar, there's actually, a couple of variants that are already either likely pathogenic or pathogenic. A couple of instances of this in one particular study. And I can already see that this study is an orofacial cleft study that actually has germline IDH2 mutation that are pathogenic as identified. But the biggest challenge is the mutations that are not necessarily annotated. So those for which we don't have already verified pathogenicity. So for this purpose, let me just take one of the genes that Hakon actually mentioned. So I'll start with, I think this was POR. Yep. And part of the reason for showing you this is actually what I do as I watch presentations like Hopkins and Nara's and Joaquin's, is dynamically actually query the genes of interest and assess them. So here, in order to review this particular gene, I can click on pathogenicity and I can see that there actually aren't any already predefined ClinVar annotations, but I can begin to review predicted features. So here we have run a number of different prediction algorithms that essentially allow you to further annotate the gene. So here, let me show you, for example, combinations of queries that one can do. So here, I'm just going to click on, for example, high VEP annotations, or secondarily choose additional mutations as predicted. And then you can combine these types of annotation and say, I want either or of these predictions and or here. And then you can assess that leveraging these predictions, there are 26 variants that meet this criteria, and then you can further review the occurrences of those. And it is not surprising that Hopkins cohort is indeed here, and that there are 6 such variants that meet these criteria that potentially underpin the results that he showed. But the part that's really exciting, and I hope more of you participate in, is that you can see these predicted damaging variants also occur across a host of other studies spanning both cancer and structural birth defects. And it's in this manner that we hope we can support the expansion of the use of each one of these data sets and allow you to come at it from a gene-centric framework. Now, for time purposes, let me just highlight a couple of other efforts that are still nascent, but begin addressing some of the challenges that especially Nara highlighted. Joaquin already essentially mentioned, the interoperability that we're driving between Kids First and the Include DCC, and this will mature and allow users to essentially seamlessly interoperate between these data sets. But really exciting are efforts that are happening across the NIH, including the Common Fund Data Ecosystem and the NIH interoperability efforts that support the kinds of a nascent effort that Nara just described. So here we're leveraging an initiative by the NIH to do exactly what Nara described, where essentially, how can a user begin to assemble data sets from multiple locations? In this context here, we're leveraging the Common Fund Data Ecosystem portal and asking for specific features, again, spanning both Kids First data sets and actually GTEx data sets that we're working with. And I'm downloading a manifest of all those data. And that manifest then contains a list of all the files of interest, and leveraging new implementations within the NIH and across Kavatica and other platforms, I can essentially upload that data and intersect it with my data. Now, the uploading really doesn't move the data. What it really does is actually point to the data in their respective sources. So just like Nara mentioned, the data often resides across multiple endpoints across the NIH landscape. And how can you assemble it rapidly is a big challenge investigators face. And leveraging these new technologies that can assemble manifests that span across these resources across the NIH allow users to immediately bring all of these data assets into a common workspace, as long as they have the appropriate dbGaP permissioning and the system already knows that those exist. So here, essentially leveraging the system itself, I, as a user who has access to GTEx located outside of Kids First, can intersect it with data sets within Kids First. And this is some of the first efforts that we hope will address some of those pain points that Nara, you had mentioned. The other exciting advancement is partnership between Kids First and the NCI and Child Cancer Data Initiative with two upcoming data sets that are essentially dually positioned across CCDI and Kids First, one on brain tumors, the other on soft tissue sarcomas. And increasingly, we're seeing the use of Kids First data sets across these platforms, like I mentioned, with GTEx and or NCI and other format. And so you'll see Kids First located across NIH ecosystem frameworks, and hope we receive feedback from you on how to make it easier and better depending on how you enter in the system. Here, for example, I'm showing the Molecular Targets platform, a newly launched platform from the NCI, where they have leveraged essentially the neuroblastoma cohorts from Kids First and GTEx data, just like I showed you momentarily, to intersect, and you can assess the specific expression level of, for example, here, GPC2 across this platform. Now, we recognize that as we try to address this complexity, we are also introducing complexity. We're introducing different resources, connecting across those resources to try and accommodate the entry point of the various stakeholders in this community. And we are continuously trying to improve and communicate about these resources and tools that allow users to connect data sets across these resources. In fact, Nara, the data set you're looking for exists within the NCI in ways that you should be able to bring into Kavatica. And we hope that this will become easier and more routine as the ecosystem mature and as cloud resources develop. But we recognize it's going to be a continued partnership between you as researchers, us as DCCs, and the stakeholders that want to empower their data. And with that, I'll end briefly and turn it over to David.
Great, thanks so much, Adam. So I see we're running a little bit short on time, so I'm gonna dive right in. And so our theme today that Marcia has been talking a lot about is collaboration. And so very quickly here at the end, I'm going to talk about, the next great collaborators here with Kids First DRC, which is hopefully all of you here in the audience. And so I just want to briefly talk about, how can we engage new collaborations? How do we make our data available, accessible for secondary use? So we've talked a lot about our Kids First data resource. And so we've released this large number of studies up to this point, which are currently available and on the Kids First portal. And of course, we still have many studies left to go. And so we're actively pursuing and already received funding to sequence and release these additional over 21,000 samples. And so we're very excited to continue our work in the future. So Marcia briefly alluded to at the very beginning, and Adam did as well, receiving access to these studies. And so every Kids First study that is released on the portal is associated with a particular study in dbGaP. And so this is how, from scrolling through the portal, how do we actually get the data? How do we get started? And so this is one of once you review the portal and find the studies you're interested in, you find the appropriate dbGaP page, and you'll see there's a little request access button right there up at the top, and that will begin your application to be able to receive access to these files. So each study has its own page. So this is the Microtia study. And so how do we get to these particular pages? So we've tried to make it as easy as possible. There's 4 different places where you can get access to these specific study pages. So one, we have a studies overview on the Kids First portal. If you click the Studies tab, it will give you a great table view of all of our different Kids First studies with links to dbGaP with their specific PHS ID. So you can click that link and it will take you right to that page to apply. Likewise, if you get all the way to the end to the file repository and you're looking at those files and you're ready to move them to Kavatica for your analysis, you can also, again, that link right there is the PHS number. And if you click that link, it will take you into dbGaP where you can apply. And then the third place is that we actually maintain a Studies and Access website, which you can access under our Resources tab, which will take you to our Studies and Access page. This provides overview information about all of our different studies, and it also includes those PHS links as well, one per study. And then finally, a great place to look if you are still stumped, you can also search on the dbGaP homepage. And so dbGaP, it provides a search box up at the top. It'll allow you to be able to find studies as well. So all of those will take you to that place where you can receive access. All of these studies have undergone clinical data review by the Kids First DRC and our investigator partners. So we're collaborating not just with our secondary users, but also, of course, with our EXO-1s as well. And so we're looking at how is the clinical data. Is it complete? Does it match our accepted values? Is it consistent across multiple fields? And so some of these studies will actually go through multiple rounds of review in order to be able to meet the quality of release that we're looking for. We also make, of course, our bioinformatics resources. And so these harmonized genomic files across each of these 4 different bioinformatics pipelines. But more than just the files, so the outputs of these different workflows, we actually make the pipelines themselves available as well. So if you have your own data set, if you have your own genomes from a separate project that you want to combine with Kids First, we make it very easy to bring your own data. And so you can bring your own genomes and combine them with the thousands of Kids First genomes in order to be able to enhance and empower your study. And these pipelines are available on Kavatica. They're preloaded and ready for use, as well as in our GitHub repository as well. And so now's a great time to bring your own data because NIH is sponsoring a cloud credits program that will support projects working on Kavatica platform. And so you can receive credits to run your analysis on research projects using Kids First data sets, for combining your own data with Kids First data sets, or for developing software tools to analyze Kids First data sets as well. So this is a great opportunity, like I said, to get started working with Kids First data. We have a website, our GitHub repo, Kids First -KF Cloud Credits, or you can do a Google search for Kids First Cloud Credits, and it will be one of the first hits to come up. And then finally, if you are still stumped and want to continue the collaboration further, we'd love to have you at our monthly office hours series. So these we hold on the second Tuesday every month at 3 o'clock Eastern time. And you can join me, members of our data team, and members of our bioinformatics group to answer any questions you have about working with Kids First platforms or data sets. So our next one is October 11th. And if you have any follow-up questions from our seminar today, we'd love to see you there. And so that is I think all that I have time for today. So I will wrap up by again acknowledging that I'm just the person here on behalf of a huge group at the Kids First DRC. We're looking forward to working with you and continuing our collaboration. So please visit us, kidsfirstdrc.org. Please follow us on social media at kidsfirstdrc, and please reach out. You can find us support@kidsfirstdrc.org by email. And so that is it for me. I'll pass it over to Marcia to wrap us up.
Thank you, David. This was great. Adam and David from the Kids First Data Resource Center, both of you kind of gave us such important information, not only learning how to access the data from the Kids First portal, but also how to find genetic variants using the tools we are building at the Data Resource Center. So with that, we already passed the time, but if I can ask you guys 1 more minute of your time, please follow up the survey about the webinar. We use that to guide our future webinars. And again, I feel humbled to be here with this amazing panel of speakers that left so amazing, like messages, really important messages. And I hope all of you are convinced today to collaborate, come and join us in this collaborative effort in the Kids First. With that, I'll see you in the spring again. We will have our fall webinar in the spring. But in the meantime, I hope to hear from you if you want to apply for one of the Kids First opportunities of support at NIH, or if you want to connect with our resources such as the sequencing centers and data resource center. With that, thank you and see you. Bye. Have a good night.